The overall goal of the following experiment is to isolate new potential intracellular pathogens present in environmental or clinical samples. This is achieved by inoculation of the sample of interest into a culture of amoebae in order to select bacteria that are able to enter and multiply in amoebae. Next, the amoebae that are lysed or in which a clear intracellular growth is observed are further analyzed in order to identify the microorganism at the species level. Then, if no bacterial growth is obtained using the Amoebal Coculture method, amoebal enrichment is performed to detect endogenous amoebae that might contain specific intracellular bacteria. Obtained results are the isolation of new bacterial species and novel viruses with this innovative method, which may be further characterized using approaches including microscopy and genome sequencing.
The main advantages of these techniques over other exist methods such as arsenic culture or mammalian cell culture is that it can be applied to isolate strict intracellular bacteria, or that you can even apply this technique to heavily contaminated samples that contain complex microbial communities. Let me introduce Sébastien Aeby, a technician from my laboratory, who will demonstrate the procedure.
To test a water sample, filter a 500 to 1000 milliliter sample through a 0.22 micrometer pore size membrane. Then, place the membrane in Page's amoeba saline, or PAS, and shake it. For solid samples such as soil, sand, or activated sludge, resuspend them in distilled water, or PBS, and filter them through a 0.